I'm Pam Soltis. I'm a curator in the Florida Museum of Natural History. And Betty asked me to talk particularly about what we can learn about Amazonian biodiversity from natural history collections. And also to tell you a little bit about the Biodiversity Institute and how that might be able to provide some resources for you guys, because I'm the director of this new institute as well. Okay, so first of all, I'd just like to mention how incredibly important our natural history collections are worldwide for helping us to understand just about all of our baseline information about natural history and distributions of species and so on. So it's estimated that there might be somewhere between 3 and 4 billion natural history specimens in collections museums around the world, and maybe about 1 to 2 billion in the US. But of course, we don't really know how many we have because until very recently, most of this information was not database, but we'll get to that in just a second. So we also have about 1,600 natural history collections in the US. And before I go any further, I think this is maybe a good time just to say how sorry we are for what happened in Rio with the Natural History Museum there having gotten up in flames. But what I've learned in the last couple of days is that the Herbarium seems to be okay because it's separate. Some of you guys may know more about this than I do, but this is what I learned yesterday. And then a lot of the vertebrate collections are also all right. So it wasn't a total loss in terms of all of their specimens and artifacts, but they had a collection of over 20 million specimens, and a good portion of those have been destroyed in addition to their research space. So there are some efforts here locally to try to figure out what we might be able to do to help the scientists there. Obviously, we can't replace the specimens, but there might be duplicates of things that could eventually be repatriated, and we might be able to help provide space or research facilities and support to the scientists. So there are some efforts going on there. If any of you guys are aware of people who have particular needs, we're really interested in learning about them, so we can reach out to them and try to help with the recovery process. Okay, so and just a comment about Herbaria, because what I didn't mention is that I'm a plant systemicist. So most of my personal emphasis is on Herbaria, and the examples that I'll give are plant related, but of course everything that I'm talking about applies more broadly as well. So worldwide, there are over 3,000 herbaria that has upwards of 400 million specimens, and these represent records of discovery over the last 400 years. Now, for the most part, we tend to think of natural history collections as being essentially the realm of systematics and taxonomy. But what I hope to share a little bit with you today is that these resources can actually provide us with a lot of additional sources of information. And some of these include the fact that the specimens themselves can provide us with materials such as materials for DNA extraction, or even genomic studies are being conducted now on tissues from specimens, chemical analyses of species interactions. You can actually make inferences about plant-herbivore relationship based on the damage patterns that you see on the leaves of a herbarium specimen. And so people are actually investigating that on massive scales now using images of herbarium specimens. And we can learn about changes in phenology, or that is the seasonal timing of events, biogeography, and lots more. But until very recently, most of these specimens have been locked away in the physical museums. So for example, here on campus, the Florida Museum of Natural History, probably to most of you guys, you might think of the building where the butterfly rainforest is. That's actually where our public programs and exhibits are housed, and some of our collections, but most of our collections are housed just right over here in Dickinson Hall, and that probably 3/4 or so of all of the 40 million specimens and artifacts that we have are actually over here in the old building in Dickinson. But thanks to digitization efforts over the last decade or so, many more of these specimens are being brought online. And when we talk about digitization, we actually mean multiple things. For a long time, people thought of digitization as simply being that databasing of the information on the specimen record, the actual label, So this would include, the scientific name, who collected it, the date of collection, where it was collected, associated species, and all of those sorts of things. And databasing that information is incredibly important because it gives us localities for the individual specimens. But more and more often now, a whole range of imaging approaches is being applied. And so, for example, there are over 20 million Herbarium specimen images in one of the databases that I'll be telling you about in just a second. So, and then we can get lots of information from the specimen images as well. So there's a rapidly expanding field of study in terms of how do we get information, functional trait information, various sorts of ecological traits off of these specimen images. So that brings me to iDigBio, and this is the National Coordinating Center for Digitization of Biodiversity Collections in the U.S. It's sponsored by the National Science Foundation, and the headquarters for this center are actually here at UF, and I'm one of the co-PIs on this project. We also have a branch over at FSU as well, so it's a collaboration between our two universities. And basically, the role of iDigBio is to help guide digitization efforts, and then to ingest the digitized information about the specimens, and then serve the data back out to the public to allow scientists and other potentially interested people to integrate the data in numerous ways. So this is a screenshot from iDigBio. If you want to check out iDigBio, there's the URL there. We currently have nearly 112 million specimen records in our database. Each of those records has maybe represents on average about 3 actual specimens, based on the fact that some organisms are have a record that goes to like a jar of fish or something like that. So we estimate that we probably have specimen records for close to 350 million specimens. We also have about 24.5 million media records, and most of these are images, and most of those images are the Herbarium specimens that I mentioned before. And so if you go to the iDigBio website and you go to the portal, you'll get these 2 pie charts, which tell you a little bit about the relative proportions of different clades of organisms that are represented in the database. So blue here is animals, green is plants, the gold is fungi, and so on. And then these are the media records, with about 82% of those 24 million being Herbarium specimens. And then if you just take a look at everything that pops up, you get a heat map that looks like this, very high density sampling in the Americas, and obviously a few other crucial locations as well. A lot of these spots out in the ocean are errors, but in fact not all of them are errors, because we do have marine organisms as well. So I thought it would be interesting as a demo just to show you what you might get for if you were to search a prominent tree in the Amazon. And so from this paper by Tristeg et al. From a few years ago, it identifies a list of the top trees, and these are the top 5. So if we actually do we can do a search for Eutrophis precatoria, which is acai, this palm. We can search for that species. There's a whole set of searching options. You can search by the genus name, the full scientific name, locality, collector, all sorts of things. You can get really carried away with it, but you can actually filter down to very specific searches if you want. So I said, let's search for acai specimens, and let's say that it has to have a map point, and it has to have an image. I think I asked for all of those. So these are the ones that that we get that have map points, and then we can actually click on one of those to get a snapshot of the specimen record, and then we can actually look at the full record if we wanted to actually look at that record. And this tells us more about that particular specimen. It also indicates that we have a photograph, and so we can look at the photograph of the image and so on. And so you can do this on, sort of an individual species basis if you just want to explore what's there. So this is all through the search portal. There's also an API so that you can actually do very large downloads and then basically collect the data into whatever application that you might be interested in. So as I mentioned, you can search by a lot of different sorts of things that might be on the specimen label, but you can also search by geography. And so you can search for a given locality, a region, or you can draw a bounding box on the map and say I want every species that's in this area, and then it'll bring you back that record as well. So iDigBio is just one of these sorts of aggregators that are pulling together information. There are a number of other national ones like this MSII in China, Canadensis, and what might be of particular interest to you guys with regard to Amazonia is Species Link, which is the English version of the Brazilian portal system. So there's a lot of information. In fact, the Brazilians were sort of way ahead of the game until a few years ago when a lot of the funding has dropped off for this. They have an excellent system with a lot of information that's been digitized. And then the Atlas of Living Australia is also a great resource just to find out how to go about using some of these specimen records. And then overall, we've got GBIF, the Global Biodiversity Information Facility. And what I'd just like to do here is just emphasize some of those resources that I think would be the most useful if you're interested in getting information about Amazonian species. So we have iDigBio, GBIF, and Species Link, and then BirdNet, which Rob was responsible for having established. And this is particularly valuable because it has actually cleaned data already, data that are usable, whereas all of these other aggregators basically take the data that come into them from the providers, and it's, you have to sort of, be careful with those data and various tools that you might want to use to clean up those data a little bit. So how can you use these specimen records? I'll just give you a couple of examples very briefly without actually describing the work. But we've been conducting ecological niche modeling for species of Florida. And on the left, you can see high species diversity and low species diversity at the current time, and then projected into the future. And you can look at how those patterns might shift. You can certainly do this for any number of regions within Amazonia, and people are doing that, not an expert on that region though. You can also use the models or the data points and associate that with a phylogenetic tree and do analyses of phylogenetic diversity. So we've done this for Florida as well, where the dark colors represent high areas, or areas of high phylogenetic diversity, essentially how much cumulative branch length from a tree, phylogenetic tree located in a particular area, and areas of low phylogenetic diversity as well. Okay, so that's my little spiel on natural history collections. And then I just wanted to end with a few comments about the Biodiversity Institute. So this was launched in 2016, or just a couple of years old. It's an inter-college institute, so we have members from all across campus, over 150 members from 8 colleges, and the Office of Research and Provost Office are providing us with our funding to get this institute going. And our 3 main goals are, very generally speaking, exploration, the use of big data and for biodiversity studies, and then finally translating this biodiversity science into action. This is where we're developing partnerships across campus in a number of different ways. But we have funding for seed grants for faculty members, some of which actually trickles into RAs for students as well. But then the other bulk of our funding goes to support graduate fellowships. And so we have academic year fellowships and summer fellowships. There's a competition each year to select the students. These are the recipients of the first year academic year fellowships. We had another 4 students last year, and we have 4 more this year. And then summer, anywhere from 3 to 5 students have been supported over the summer. And I just wanted to note a couple of examples of Amazonian ones. I put a little asterisk there because in fact, on a study is on the Atlantic forest, but it seems so relevant to questions about the Amazon as well. I thought it was that too. But we also have a number of other students who are working in the Andes and who either had summer fellowships or their academic year fellowships funded to help with that work as well. So a lot of different types of studies that have been supported already through the Biodiversity Institute. So I'll encourage you guys to put some applications later on there. That's all I have. I don't know how I did on time. Hopefully not too long.